Hi everyone, today we're going to continue the fat metabolism. I am Tommy. I'm going to explain to you today the triglyceride breakdown, the fat mobilization, the thing that everyone wants. So let's see the metabolism behind that. Let's go. So if you remember from the last video, we got the triglyceride formed and we said that it was stored into the adipocytes. Okay, so let's have a look to an adipocyte. This is an adipocyte, a fat cell. What we need to know here is that they are inside of these vesicles, and there are many triglycerides here. What we're going to do is break them down and create, once again, glycerol and fatty acids. So how are we going to do that? Well, the conversion of triglycerides into fatty acids and glycerol happens when we are not eating, when insulin is low, and when other hormones are activated, like glucagon, adrenaline, and noradrenaline. These hormones are called hyperglucemic hormones because they increase the blood glucose levels and they stimulate the sympathetic system, like adrenaline and noradrenaline. So there is a very meaningful reason behind that, because free fatty acids then can be used as energy to the muscle when we are in a fight or flight state. So to begin with, we have a G-coupled protein receptor here. These hormones are going to bind to these receptors and the G protein, and then they're going to stimulate the adenine cyclase to create cyclic AMP from ATP. So this enzyme is converting ATP to cyclic AMP. Cyclic AMP is then going to stimulate the protein kinase A. If you remember, we already discussed this process in the glycogen metabolism. These hormones are doing this pathway every time. Glucagon indeed is stimulating protein kinase A. We already discussed that in the fatty acid synthesis video as well. So we have protein kinase A with the phosphates, and it's going to put phosphates on very specific molecules we have here. In the vesicle membrane, we have the CGI bound to the perlipin. These are together, but when the protein kinase A acts on the perlipin, putting phosphates on him, they're going to separate. And once they separate, CGI is going to activate the adipose triglyceride lipase. Okay, adipose is where we are in the adipocytes or adipose tissue. Triglyceride is what we have here. Lipase. Lipases are enzymes that cut down lipids. So we are going to cut, that's why we have a scissor here. We are going to cut one fatty acid off the glycerol, and we get with a fatty acid, free fatty acid here, and the diglyceride. Then the protein kinase A is going to phosphorylate the hormone sensitive lipase. As being hormone sensitive is triggered by the hormone binding site, which indeed activates the protein kinase A, so that's why it's hormone sensitive. And this lipase is going to cut another free fatty acid of the glycerol. And we are left with a monoglycerate here. And the third enzyme involved in this process is the monoglycerate lipase. And once again, protein kinase A is going to phosphorylate this enzyme. From the breakdown of the monoglycerate, we get glycerol and the third free fatty acid. And this was the triglyceride mobilization, the fatty acid creation from the triglyceride in the adipocytes. I hope you understood it well. Hope you enjoyed. Subscribe to the channel to help me. Like the video so it will spread to more people. Share it if you want to your friends and maybe classmates who need this. And this diagram is going to be down below in the description so you can download it and study it very well. I see you on the next one.